Well, thank you so much for being here. Thanks for the presentation. It's a great honor to be here presenting a little bit of our work and sharing some of our results. So as already mentioned, my talk will be related with mammal functional diversity, specifically exploring patterns, trends, the influence of human intervention, and then how we apply that into local conservation or on-the-ground conservation. So first, just as part of the context, we are facing a critical moment for biodiversity. The last global mammal assessment said we have 25% of mammals threatened with extinction globally. And so far ecology has been, and macroecology, one part of ecology, has been focusing on understanding patterns and processes that explain diversity and diversity patterns across the globe, even from the 50s and 60s. But now it's not only about explaining diversity, but because we have so many mammals threatened, and probably even more in the more recent update of the assessment, we now need to understand patterns that explain extinction, the drivers of extinction, and the patterns of threats distribution. This in order to understand what's going on. The most recent efforts to understand or to study the global biodiversity crisis, it's showing that the crisis is even bigger than we thought before. Species are going extinct at least at 100 times faster than the normal rates of extinction globally. So that means we are facing something that it will affect us on the short term. Part of the analysis that have done so far include global conservation priorities, which is identifying those areas that require attention more urgently so we can prioritize and invest scarce resources so we can ensure we maintain these species on the long term. One of these efforts was in 2008, which was precisely the global mammal assessment, where we understand for the first time for all mammals across the globe how many were threatened, what was the distribution. This is actually the map of species richness globally. And what was going on exactly with mammals. What are the main threats, how are they distributed, how are they affecting many different groups. This allowed to do conservation planning and link from global analysis and macroecology down to local scale. Conservation most of the time occur not at the global scale, but on precise locations where you make decisions that change the fate of a species. So all these progresses have allowed to better understand where we should focus efforts and work towards ensuring as many species as possible. However, until recently, we didn't have the information at the appropriate resolution so we could move from these scales, from global to local, because most of the information was not available, we didn't have the tools, etc. But right now, we have precisely all the information we need to start working on understanding patterns and how these patterns move across the scales. Also, recent advances on how we measure diversity have allowed us to understand better how species interact and what means in species within an ecosystem. One of these is functional diversity, which is defined as the diversity of ecological traits and life history traits defined on space and time. But most importantly is because that allows us to understand what species role plays on what roles the species play on ecosystems. What are the relationship between them, how they influence ecosystem processes, and of course at the end, how ecosystems function and how they provide the services they provide and how we can maintain them. The main goal of functional diversity is to understand what processes determine the persistence, resilience, and functionality of ecosystems. And that is probably one of the best tools, it was developed for plants. More recently, it's been explored for animals. But that could allow us to do better on-the-ground conservation planning in terms not only on protecting species or protecting ecosystems, but also protecting evolutionary history if we think of phylogenetic diversity, but also how species play roles in ecosystems and how we can maintain those processes. So in general, I'm going to present some different projects that we're working on, all related, but the general framework of the concept is to analyze species diversity on different measures, on different scales, from global to regional and national scales, trying to understand or exploring patterns and priority selection. This means only to understand the patterns, like a description of how it's distributed, where and why, and then select those areas where we should focus efforts in order to maintain them. That includes understanding extinction risk as part of the ecological system, because of course if we're thinking that at least 25% of species are endangered or threatened, we need to understand what's the role those species play, and then see how big is the problem. Also, to understand determinants, or mostly drivers, of these patterns, and what are the current conservation efforts, and like how are they performing in terms of maintaining this. All of these just to go back down to underground conservation. The main idea is if we can understand global patterns, we can then take down the scale and see how this fits, like local scales where conservation occur, how they fit into the larger picture. So first we built a database including taxonomy, ecology. This ecological part of the database included functional traits, which are defined as ecological or life traits related with the species, body mass, habits, trophic yield, etc. And two spatial databases, one related with the species distribution that was obtained from the IUCN Red List of Threatened Species, and then one spatial in order to understand ecosystem functioning. So we use WWF's ecoregional regionalization of the world, including ecozones, biomes, and ecoregions. And from there, we obviously use different measures, species richness, which is the most basic and the most used for understanding global patterns. But then we included functional diversity and functional groupings, two different measures of functional diversity used in plants, but obviously adapted to mammals. And we use spatially explicit or geographically weighted generalized linear models in order to understand what's the relationship between drivers and these patterns and how they vary in the space. So this is just to show part of the context. This is the map of species diversity, species richness on the top, and then down phylogenetic diversity that was derived from the Global Mammal Assessment in 2008. Just showing obviously what we all know about the diversity latitudinal gradient with more species concentrated towards the tropics, which also is better related with phylogenetic diversity towards the tropics. So now we look at functional diversity, and the pattern is obviously very similar. We have a large concentration of functional diversity or variety in life traits in the same place for the tropical areas and also for the functional groupings. That means that there are more different groupings in tropical areas than in temperate areas. And it's consistent with the latitudinal gradient. So we expect to have more or higher functional diversity values towards the tropics, but it varies according to the ecological zone. That means the Afrotropical and Neotropical area, it's obviously concentrated towards the middle, and then we have the Palearctic and Nearctic on both sides, on both tails of the distribution. But more interestingly, what we found is that according to the world's ecoregions, functional diversity behaves quite similar in similar environments, even if they're totally unrelated in different locations and with different species composition. If you see these two clusters, the first one is related with ecozones, where if you see the significant clusters include Indo-Malayan, Neotropical, and Afrotropic. That means that not even considering species composition, like species lineages and different evolutionary histories, we will find similar values or similar levels of functional diversity in these areas. So you see two groupings, the tropical areas and the Arctic areas. And also it's the same for major habitat types, for biomes, which is the lower cluster. Oh, sorry. Well, what we see here is that tropical ecosystems, which have very similar structure, including dry, mountain, and tropical basal ecosystems, have very similar values of functional diversity. And it's the same for grasslands, and it's the same for temperate ecosystems. So this somehow shows some congruence, or how different species, with different stories, come to the same habitat and play similar roles. Obviously, we also could identify where these high values occur. Interestingly, is not only the tropics, but we have like these hotspots of high functional diversity even in temperate areas. And that means those areas where we have many different species that play many different roles, and are quite different to the other areas. And we can see some hotspots here in Asia and Russia. So we started exploring some of the most basic relationships, including macroecological relationship, but also the most basic, which is how functional diversity behaves with species richness. And obviously both measures are very highly related, but I'm going to show you how spatially that is not necessarily true. But in terms of values, they are quite related. And with area, interestingly, it's also, they're highly related, but it varies according to the ecozone. So the neotropical and aphrotropical areas increase the value of functional diversity as you increase the area. But it's not the same intercept, not even the same slope for other of the areas like the Palearctic, the Antarctic, etc. So here we also see the difference between Arctic, like temperate and tropical ecosystems, and how all tropical ecosystems, independent of the location, behave quite similar in terms of these macroecological patterns. But also, even if we have this very linear and clear relationship, mismatch occurs, which is where species richness not necessarily explain the entire variation of functional diversity. And we find, especially in Africa, we find these hotspots where the relationship is not completely linear. And it's the same for area, especially Africa, and in this case, Indonesia and Asia and Oceania are very interesting in terms of area, and we all know the high diversity we have in these areas, and is not completely related with area like we have in the neotropics. So the next step, sorry, that's in Spanish. The next step was to understand what species at risk, what's the role they play in functional diversity. And that's important for us because those species that are threatened are obviously more prone to disappear on the short term. And if we lose those species, we're going to lose a part of that functional diversity. The idea was to understand how much of that. So we explored it to all the ecoregions, and we found that there are some areas, for example, in Africa, where only 11% to 18% of the variation of functional diversity is explained by threatened species. But then we have areas like the neotropical area and the Far East, where even to 80% of the functional diversity is explained by threatened species. And this obviously, when we extrapolate to ecoregions and biomes, behaves quite similar. But this is one of the most interesting results for me because what this says is that we are eventually facing a loss of functions, a change in the ecosystems and how they work, and obviously the implications of ecosystem services, etc. But pretty much how the ecosystem works is going to change if we lose all these threatened species. The effect is obviously spatially differential, so endangered species, for example, are very important for North America, for the Nearctic region, while data deficient species are very important in general for America. So this means, for example, the areas that are highlighted for critical endangered are even more important because these are the species that are more likely to disappear in a very short term. But anyway, any of the 3 or 4 categories, these areas are the more likely to suffer changes as we know these ecosystems right now. So again, as I told you at the beginning, what we are doing is trying to downscale these to a level that we can actually do some real conservation. So the first step was to take it down to the neotropical realm, which is obviously, there's a bias there, where we map species richness, functional diversity. We explore how biomes behave in terms of these 2 measures. And again, we found like tropical and subtropical mountain, coniferous, dry, and moist forests behave the same. And they cluster the different types of ecosystems in terms of these 2 measures, especially functional diversity. And then we explore what's happening with intervention, how human intervention measure as the change of land use by humans could affect functional diversity, and how species at risk affect functional diversity. So we found, for example, very high influence of intervention in northern South America and northern Mexico, and very high influence of threatened species for the Guianan shield and southern Mesoamerica. All these just to have maps like these, which can allow us to say like where we should focus or we should prioritize efforts in order to reduce the potential loss of functions or ecosystem functioning for these species. So this is the risk. This is measured as what's the risk of losing these species and how much the functional diversity would change. And we already see Caatinga, we see Yucatan Peninsula, northern Colombia, Costa Rica, Panama, where we're going to have very high risk of, if we lose these species, we're going to suffer a big change in functional diversity. And the expected, which is if we don't lose those, still what's the risk, you know, like if we don't lose all these species. And then we have the Guianan Chill and northern Colombia, southern Mesoamerica, where the risk is more high if we don't do anything to retain these species in the landscapes. So we downscale again, and then take a closer look, this time to Costa Rica, and see how it behaves. And at this scale, one of the, because of the resolution, we could explore deeper into what are the main drivers of these patterns, not only at looking at the pattern, but try to understand a little bit. So this is the species richness, this is functional diversity, this is the distribution for Costa Rica. You can see there are some clear differences. So first, for species richness, we found that mostly environmental variables are the drivers of the species richness distribution, and some anthropogenic variables. Interestingly, human density and human appropriation of primary productivity. But again, we have regions where we cannot explain species richness with these variables. I mean, 3% of the variation, but some areas, 43% of the variation is explained. So we did the same thing, but now with functional diversity, but then we saw that functional diversity, it doesn't behave the same globally than locally, and the relationship with the species richness, which was expected at all scales, is not necessarily the same. And this is, of course, because we have, in a country like Costa Rica, we have a very, very clear dominance of two groups, obviously bats and rodents. So we map what's the relationship between these measures, and this is one of the mismatches I mentioned before of the two measures globally. And we had clearly identified these three regions that behave quite similarly and are explained by these two groups. So we had to explore functional diversity on these three regions and not for the entire country. So I'm not going to go into numbers, but these are the variables for each of the regions. And the most important part is that some areas like northern Costa Rica are not very explained for these variables, but still it's interesting. One of the main variables that explain functional diversity in northern Costa Rica, which is the dry forest of Costa Rica, is the origin of the species present in the area. Origin understood if it's of northern or southern origin. And the dry forest, which is obviously Costa Rica is the southern limit, is just sharing functional diversity with the rest of the dry forest. But the other two, which include the mountain range and the Caribbean plains, it's really, really well explained by local variables and environmental variables, such as the ones I show you. The same exercise was done for Colombia, trying to understand, like these patterns, how they work. In this case, the approach for Colombia was to understand where was the mismatch. And I'm going further into that, but one of the things is that most conservation and conservation priority definition is based on species richness, which is the most basic measure. But what we found is that species richness, phylogenetic diversity, and functional diversity are not necessarily homogeneously distributed in the landscape. So if we use only one measure, we're missing a piece of the puzzle. So in this case, we measure mismatch, and what we found is that there are some areas where one measure cannot be like a surrogate of the other two. And the thing is that if we are thinking on real conservation or comprehensive conservation, we need to think not only in species richness, but we need to think on protecting or saving evolutionary history. And of course, we need to save functions, and we need to save how ecosystems work. So now that we know we have these patterns, the next step was to evaluate if the current conservation efforts are working. This is analysis of effectivity of protected areas for protecting species richness and range of all mammals in Costa Rica. Here's the evolution on how the protected area system grew in time for the country. And we found that even when the system is really good by protecting a large number of species, at least 98% of the species are present at least in one area, we still have very large hotspots of diversity that are not currently covered and that require attention in order to maintain mammal diversity in the country. So just as a general discussion, and I'm going later into more details of how we apply this. So part of the main goals of this work is trying to understand, and it's been one of the goals of ecology for a long time, is to understand distribution patterns of diversity and what are the underlying reasons explaining these variables. But the thing is that it's not a straightforward answer, and one of the main conclusions is that when we moved across the scales, variables started to change, patterns started to change. So there's a big deal here how we can link the different scales, because most of the global priorities definition and where big or large amounts of money are focused are based on global analysis, which is obviously fine. But if we look closer and we use other measures, not necessarily we're being efficient on how we allocate those resources. Also, diversity gradients, which was expected, very classic works, but it works the same for functional diversity too, which is very, very interesting. And the recent advances, including diversity ecology, which is not only, it's also understanding measurement components and interactions, I think can provide more details on not only understanding the ecosystem and diversity per se, but also how to protect those. Not only working with species and ecosystem, which can be like, like a recent discussion that what we should protect, but protecting the entire ecosystem and protecting the functions that occur in there. And I think it's very important to clarify that not only to study these patterns, not only understand variation, but how ecosystem functioning and historical processes operate right now, and how explain those patterns, and how can we work to maintain them. In terms of patterns, well, mammal functional diversity, it's very highly related with scale and species richness, but there are also multiple drivers that can explain this. And one of the main drivers, or not drivers, but explanations, is ecosystem structure. So one of the things is that these clusters I showed you were independent of the location, independent of the species composition, functional diversity tends to behave the same in different locations across the world. So that is one clue that we could move forward between scale. We just need to find the appropriate variables, which is part of what we're doing now. Two main hypotheses rise from these patterns. The first one is environmental filtering, so how species assemblages are constructed. Can be one of the explanations on why we find these diversity patterns and these congruence. And then also ecological trace convergence, which means like species with completely different evolutionary lineages play very similar roles in similar ecosystems in different locations. So that's quite interesting. Ecological convergence has been studied quite well across the world, but now to understand not only in terms of ecological features, but the functions per se in the ecosystems, it's even very, very interesting. And another interesting thing is how the species-area relationship work. I mean, there's a lot of works into this topic, but according to results, also it can vary according to ecological traits. That means the effect of area is not only on the number of species, but plays a special filter, in a way to say, on specific traits. And it depends totally on the ecological context. That means that we cannot analyze the species-area relationship across the world, but we need to see the details on how heterogeneously they distribute across the globe. And in terms of conservation, functional diversity is ecological traits complementary within assemblages. The thing here is that the species composition related with, together with vulnerability, means also the risk of functional loss and resilience of ecosystems. So this is the link between species and ecosystem processes. There's a strong effect of endangered species over functional diversity, which is very interesting. In general, most endangered species are rare or restricted with small distributions, but apparently they play very important and complementary functions in ecosystems. And at least, apparently, average in the world, 60% of FD of functional diversity is influenced by species at risk. So that's a big deal because we're talking about 60% of the ecoregions of the world are probably facing loss of functions and therefore services and ecosystem processes. Even certain ecoregions are 80% determine functional diversity by species at risk. So that means vulnerability of the functional stability, or at least as we know ecosystems right now. And the thing here and the key concept is that if we lose a species, we lose functions. And that's a big thing and a link that probably could help us for policy making, among other stuff. And quickly, just to finish up, our application of this goes then to local functional landscapes. So it's not only to understand global patterns, not only to understand it at national levels, but to see how they behave at local scales. So what we think is that connected landscapes plus ecosystem restoration should be equal to ecological resilience. So what we're doing is functional landscapes where we obviously need to connect, we need to restore, recover, and conciliate. Obviously, conservation is a human topic. And for doing this, we've been working trying to understand where are the hotspots, what's the distribution of many different species. In this case, for example, this is an exercise for Costa Rica, Colombia, and Mexico of jaguars. Where are jaguars located? Obviously, using a little bit of this charismatic umbrella species. And from there, we select, you can see in the map, the areas we highlight here, here, and here. And from there, what we're doing right now is exploring specifically these areas, how to connect them, and most importantly, the next question is how functional diversity behaves at local scales within the ecosystem, and what's the heterogeneity, and all those clues that we can then inform so we can move forward again back into the other scale and see if we can match these measures. Some of the initiatives we use for that is, for example, and I'm presenting this to you, the Jaguar-friendly certification, which is production of agricultural products using Jaguar as a flagship so they can sell better the products and they maintain ecosystems, environmental services payments, education, and sustainable production. This was just like to give an idea of what we're doing. And that's it. Thank you.
Jose managed to squeeze all that into his allotted time.
Sorry, yes.
He's been busy. Questions for Jose? Nobody has questions? Yes, Nate.
Could you talk a little bit more about how you calculate functional diversity?
Sorry, say again?
About how you calculate functional diversity, including like the traits that were involved in it?
Okay.
Sorry, Jose, do you want to repeat the question just so that...
Okay.
Yeah.
How did I measure functional diversity using the traits? So what we did was using the distribution maps for all species, we estimated which species were present on the different ecoregions. From that species list, together with the database of life traits and ecological traits, we use different indexes. We stay with one index, which is Pechis and Gaston. It's a dendrogram that connects all species into a branch. It's very similar to the one that used for phylogenetic diversity. And functional grouping. So two measures is just two index that try to connect all species present on that place based on the ecological traits of all species.
Other questions? Well, I'll ask one. So you're the recipient of our Hornaday Award. For students interested in pursuing conservation and conservation themes, if you had to give them one piece of professional advice, what would you recommend they study or do or engage in to promote that aspect in their career?
I think the most important thing is to understand the needs. You know, like, I mean, as many conservation scientists just stay on the science part and you know, like we can answer many questions, but then reality is a different stuff. And we know how species are being lost, but we don't know how to save them. So I think we need to link those two and really understand what are the needs, where we should focus research efforts, and most importantly, move to action too. You know, like we cannot stay only from the academy watching how we lost species.
Mm-hmm. Okay. Any last question? Yes, Rudy.
How well are you able to translate your elegant work to the political level? And what's the bottom line? When you talk to leaders and ask them to do these things, how effective are you? I mean, what do
I think it can be done. It is quite difficult to go to decision makers and politics and explain functional diversity. But we can explain the potential effects that we're gonna get if we lose or we keep losing species. And then obviously it's easier to make clear to people, to the public in general. One of the things, for example, with functional diversity is that when we talk about ecosystem services, water, air, pollination, et cetera, et cetera, et cetera, one of the clear things that they depend that with the ecosystem is working properly, and for the ecosystem working properly, we need the species. So what we're doing, for example, in Colombia and Costa Rica is working with the government to ensure we have a more comprehensive protected area system, just focusing on saving as many species as possible. And from there, we work more with economic tools in terms of how we value ecosystem services and linking that into basic ecology of species.